Well, hello again and welcome back, pre-meds. I hope you're all pushing through with that MCAT content review. Today we're going to be going over carbohydrate structure and function, specifically monosaccharides. So this is one of two on carbohydrate structure and function. The next part will include disaccharides and polysaccharides. So let us get started over here. Don't gatekeep these videos. If you have a friend who's currently studying for the MCAT, be sure to share the playlist with them. It really helps the channel grow and I would really appreciate it. So we're going to start off by talking briefly about the naming system. Not extremely high yield, but you definitely do need to be familiar with it. So the names typically start with the Latin prefix for the number followed by the suffix "-os". So for example, triose if you have 3 carbon atoms, tetros, pentos, and hexose. Now how about diose if you have 2? Well, turns out you can't have less than 3 carbon atoms in a carbohydrate. So the most simple form, for example, would be glyceraldehyde, and that contains 3 carbon atoms. Now when it comes to carbohydrates, some will have an aldehyde and others will have a ketone. So for example, glucose has an aldehyde in it, whereas fructose has a ketone. If it has a ketone, then it's aptly named a ketose, and if it has an aldehyde, then it's called an aldose. All right, so for example, you can have an aldotriose where you have an aldehyde and 3 carbon atoms within the carbohydrate. And then you can have, for example, a ketohexose. So an example of that would be fructose, where you have a ketone and 6 carbon atoms within the structure. Now, sometimes the MCAT will use common names instead of these, like this fancy naming system. So for example, glucose, fructose, and mannose are here just named by their common names. And just note over here how these are aldoses because they have an aldehyde, whereas fructose is a ketose because it has a ketone over there. Okay, let's move on from naming to stereochemistry of carbohydrates. And this part over here is extremely high yield, so please pay very close attention. So let's first discuss the D/L classification versus R/S absolute configuration. Now it's very important that you know that these two things aren't interchangeable. Like there's no rule that says, for example, if a molecule is a D molecule, then it'll necessarily be an R or S molecule, and same thing for L molecules. Even for amino acids, where there is sort of a rule with some exceptions, you really can't use them interchangeably, and it's sort of on a molecule by molecule basis. In the case of carbohydrates, when we're discussing carbohydrates, we rarely refer to the R/S absolute configuration. We focus more on the D/L classification. So how do we assign D or L to a carbohydrate? Well, it's all based on the glyceraldehyde molecule over here. So this is glyceraldehyde. And first of all, you're gonna have it on the Fischer projection. So this only works if you have the Fischer projection. So you identify your chiral center, and by the way, you have to go with your very last chiral center. But in the case of glyceraldehyde, it really doesn't matter because it's your one and only chiral center. So you go with that one, and then you take a look at the highest priority group attached to it. In this case, it's OH. If that OH group is to the right, then you have yourself a D glyceraldehyde, and if it's to the left, then you have an L glyceraldehyde. So let's move on to glucose over here and see how we can identify that. Well, here again, you're going to look all the way to the bottom, so you count all the way until you reach your very last chiral center, which is this one, and you check if the OH is to the right or to the left, and you follow the same rule over there. All right, so it's important to note that these two molecules are stereoisomers relative to one another, and these two are also stereoisomers. Well, what does it mean for two molecules to be stereoisomers? Essentially, they have the same chemical formula, they just differ in how they rotate plane polarized light. Now, more simply, they just differ in one or more chiral center. That's all a stereocenter is. So you have at least one chiral center where they have the groups flipped. So in the case of glyceraldehyde, the OH and the H are flipped in L-glyceraldehyde versus D-glyceraldehyde. And then in the case of D-glucose versus L-glucose, you can see that all of the stereocenters or all of the chiral centers are flipped. And there's a special name for when two stereoisomers have all of their chiral centers flipped, and that is enantiomers. So enantiomers are a special type of stereoisomers where they are non-superimposable mirror images of one another, and we'll see that what that means exactly on the next slide. But essentially, you want to know that all chiral centers should be flipped. So in the case of D-glucose and L-glucose, you can take a moment and you'll see that anywhere you have OH on the left in D-glucose, you have it on the right in L-glucose, and likewise for hydrogen. All right, so now let's go over this slide just to really hammer down that enantiomer point, or non-superimposable mirror images point. So you can see over here, we have D-glucose, L-glucose, and also another D-glucose molecule. Now, what happens if I pick up this glucose molecule and try to superimpose it on top of the L-glucose? Well, it doesn't really fit neatly. Like you can see that OHs are being stacked up on top of hydrogen atoms, and hydrogen atoms are being stacked up on top of OH groups. And that's because they are enantiomers, so they're non-superimposable mirror images. However, if you pick up the D-glucose molecule and try to superimpose it onto this other D-glucose molecule, you can see that they fit perfectly, like they overlap perfectly, because these two are the same exact molecule, whereas L-glucose is very similar, it's just that it's flipped on every single one of those chiral centers. So it turns out we have another type of stereoisomers, specifically called diastomers. Now, diastomers can only exist when you have more than one chiral center. So glyceraldehyde, you only have two stereoisomers, and they are enantiomers of one another. But because you only have one chiral center, you don't have any diastomers. Whereas if you look at the structure of glucose right here, you can see that you have 1, 2, 3, 4 chiral centers. Remember, this one isn't a chiral center because it's attached to 2 hydrogen atoms. So we have 4 chiral centers. So that means you can have diastomers of glucose. And indeed, this is a diastomer of glucose, which we call mannose. Now, notice the difference between mannose and glucose. The only difference is in this hydroxide group right here. So you can see in glucose, that very first one on that first chiral center is pointing towards the right, whereas on D-mannose, it's pointing towards the left. That's the only difference. The chiral center is flipped, that first chiral center. So these are what we call diastomers. All right, now it doesn't just have to be one chiral center that's different. It can be one, in the case of glucose, 1, 2, or 3. But importantly, it can't be the case that all the chiral centers are flipped, because then we wouldn't call it a diastomer. Rather, it'll be an enantiomer. So you can see that when they're all flipped, you go from D-glucose to L-glucose, and that's simply an enantiomer and not a diastomer. Okay, now importantly, when you look at D-mannose, it's also called D instead of L. And why is that? Well, if you look at that very last hydroxide group, it's pointing towards the right. And when it's pointing to the right, we're going to call it D-mannose. Okay, now to calculate the total number of possible stereocenters, not the possible diastomers or the possible enantiomers, the possible stereoisomers, you're going to use the equation 2, very simple equation, 2 to the nth power, where n represents the number of stereocenters that you have. So in the case of glucose, it's going to be 2 to the 4th power, so that's just 2 times 2 times 2 times 2, which equals 16. So you have 16 stereoisomers of glucose. And now, this might seem a bit counterintuitive, like we mentioned that you can have, okay, you have enantiomers where they're all flipped, and then you have diastomers where you can flip 1, 2, or 3. How did we get 16? Well, it really lies in which ones you decide to flip. So for example, when we say we're going to flip 2 chiral centers, well, it could be the case that you flip the first and second one, or the first and third one, or the first and fourth one, or the second and fourth one, or the first and third one. So you can see how those diastomers really start to pile up. So in the case of D-glucose, it will have 16 stereoisomers total. 15 of them will be diastomers, whereas only 1 will be an enantiomer, where all the stereocenters have completely flipped. Okay, let's take a look now at cyclic sugar molecules. This is also extremely high yield. So, you know, sugars contain aldehydes, ketones, and hydroxyl groups. So like any other organic molecule, they are going to undergo the organic reactions that we learned in organic chemistry. So the sugar itself, or glucose itself, can undergo what we call an intramolecular reaction, where you have a hydroxyl group, which is a nucleophile, and then you have a carbonyl group. Well, that carbon of the carbonyl group is an electrophile. So you can have a nucleophilic attack where this very last hydroxyl group attacks the carbonyl carbon, or in the case of fructose, for example, it'll be a carbonyl carbon, a part of the ketone functional group. In this case, it's an aldehyde functional group. In either case, you're going to get a cyclic ring over here. So notice what's happening here. This oxygen attacked the carbon of the carbonyl. All right, and then what we call this is a hemiacetal. A hemiacetal, basically called a hemiacetal because you have an aldehyde. If it was a ketone, in the case of fructose, you would have a hemiketal. Okay, also just a lot of terms over here. This is called a pyranose because it has it's a six-membered ring. If it were five, such as in the case of fructose, it would be called a furanose. And we'll get to why, even though fructose has 6 carbon atoms, why it ends up with a 5-membered ring. We'll talk about that in a second. Now, it's important to be able to convert between the Fischer projection and the Haworth projection, because sometimes they'll directly ask you, like they'll show you a Fischer projection and tell you to choose the correct Haworth projection, and they'll look very similar, like the 4 options of Haworth projections. So you need to really understand how to draw this out. So what's going on here is you take your oxygen, so that's going to be the very first one over here, the first member of the ring, let's say. The next one is going to be the carbon of your carbonyl. So that's this red one over here. I've color coded it so it can be as clear as possible. Now the next carbon atom is going to be this green one over here, and then the blue one, and then the black one, and then the gray one. And then finally you have this group attaching out, which is CH2OH, which is right here. Now notice one thing, kind of an aside, not very relevant to the point, but notice how this carbonyl is now a stereocenter. So previously it had a double bond with oxygen, it wasn't a stereocenter. Well, now it's attached to 4 unique groups, so it is a stereocenter, but that's not very relevant to our discussion. Now that's drawing the ring. Now the more important part is how are you going to know whether these OH groups are pointing up or down, or the hydrogen atoms are pointing up or down? Well, there's a very simple rule. If on your Fischer projection the groups are pointing towards the left, then they're going to be going up on the Haworth projection. If they're going towards the right, they're going to be going down. So over here you can see how that very first carbon atom, this green one, has hydroxide going towards the right, so it'll be going down. And then the next one has hydroxide pointing towards the left, so the hydroxide will be going up, and so on and so forth. Okay, now I did want to comment very briefly on why fructose, even though it has 6 carbon atoms, so it has the same chemical formula, C6H12O6, as glucose, but it ends up with a 5-membered ring. Very quickly, I just wanted to highlight why that's the case. So we have fructose right here. So notice that because fructose has a ketone, when this hydroxyl group attacks the carbonyl carbon right here, what you're going to have is, so remember, we mentioned that the very first member of that ring is going to be the oxygen, and then your carbonyl carbon, so that's 1, 2, 3, 4, and 5. So you have 5 total members because the ketone is sandwiched between 2 carbon atoms, whereas when we talked about glucose and it attacked the aldehyde, the carbonyl was at the very end of the molecule because that's how the nature of aldehydes are. Okay, now let's move on and talk about chair conformations, which also show up on the MCAT. Now don't worry, we're not going to get into too much detail with chair conformations. We'll talk a lot more about them in our organic chemistry playlist, but here we're going to talk to them as they relate to the topic at hand. So the chair conformation, they're just a way that cyclohexanes, when you have a cyclic 6-membered ring, it arranges itself just naturally into a chair conformation. It's just more stable. And in the case of carbohydrates, those groups are either going to be a hydroxyl group or a hydrogen atom, right? Because if you look at the structure of glucose right here, all the atoms are attached, the carbon atoms are attached to either a hydroxyl group or a hydrogen atom. So what you want to know when it comes to the chair conformation is that you have the groups are either going to be in the axial, this is the axial position, or the equatorial. This R right here is the equatorial position. And importantly, you'll notice that the axial and equatorial positions alternate throughout the ring. What does this mean? So notice how right here, this hydrogen, which is on the axial position, so it's vertically pointing up, okay, at a 90-degree angle. Whereas if you look at the R group, the equatorial position, it's going to the side and down, right? So kind of horizontally, but pointing downwards. Well, on the next carbon, the next member of the ring, it has to be that the one in the equatorial position, again, in this case, the R group, is going to be pointing up. So even though it's still going to the side, it's in the equatorial position, that's what it means to be going to the side, now it's pointing upwards. And now the one that was previously pointing up, which was the axial position, is going to switch to be pointing downwards. So again, just to really hammer this point, both axial and equatorial substituents can either point up or down. But the idea is that axial ones will point vertically up or vertically down, and equatorial ones will point to the side, and they'll either be going towards the top or the bottom. Okay? And very importantly, the bulkier group always tends to be pointing in the equatorial direction. So because that reduces steric hindrance, so regardless whether the equatorial position is going towards the top or towards the bottom, that bulky hydroxyl group relative to the hydrogen atom is going to prefer to be in the equatorial position. Well, let's take a look at D-glucose in this case and in this case. What's the difference? The only difference, they're pretty much identical. The only difference is this carbon right here, which we call the anomeric carbon. This anomeric carbon can either have the hydroxyl group in the axial position or the equatorial position. If it's in the equatorial position, we call it the beta-D-glucose molecule. If it's in the axial, we call it the alpha-D-glucose molecule. Now, the beta-D-glucose is going to be more stable, right? Because the bulky hydroxyl group relative to the hydrogen atom is in the equatorial position rather than the axial position. Okay, now we have something called mutarotation. Essentially what this is saying, I know this seems like a lot, but it's a pretty straightforward concept. When you have, let's say you have a beta-D-glucose in solution in water, what's constantly happening is that this ring is opening and closing, as you can see right here. So it's opening and closing. Now, once it opens, it can sort of decide whether it wants to revert back to the beta-D-glucose when it closes or become the alpha-D-glucose, because these atoms or these groups can be rearranged so that you shift to the alpha-D-glucose. Now again, we said that beta-D-glucose is more stable, but it's not that much more stable to the point where you won't have any alpha-D-glucose. As a matter of fact, in solution, you'll typically have about like 30-35% alpha-D-glucose, and the remainder, the majority, will be in the beta-D-glucose because it is indeed more stable. Okay, now let's talk about the chemical reactivity of monosaccharides. So recall earlier I mentioned that carbohydrates have these functional groups, aldehydes, ketones, and alcohols, and therefore they'll undergo the organic chemistry reactions that these functional groups undergo, right? It makes sense. Okay, so we're going to talk about oxidation reduction reactions and esterification reactions. We're going to leave glycosidic bond formation for when we talk about disaccharides and polysaccharides in the next slide deck. Okay, so when it comes to oxidation reduction reactions, it's very important to recognize that the aldose sugars, like glucose for example, because they contain an aldehyde, they can be further oxidized into a carboxylic acid. Now recall when you're thinking about ketones from organic chemistry, you sort of run into a wall. You can't further oxidize a ketone. So they shouldn't be reducing sugars, whereas the aldose sugars are reducing sugars because essentially they can be oxidized, and when they themselves are oxidized, then they're acting as the reducing agent, and that's why we refer to them as reducing sugars. And in fact, there are reagents that we can use to test a sample and see if it contains one of these reducing sugars or not. And as you may have predicted, these tests are going to rely on oxidizing agents. So essentially you're going to put an oxidizing agent in your sample, and if it tests positive, then you know that you have a reducing sugar in that sample. The first of these tests is Tollin's reagent. Tollin's reagent relies on this silver-containing compound. What you want to know is if you get a positive test, you're going to see a metallic silver color, and then you know that you have a reducing sugar there. Or you can alternatively use Benedict's reagent, which relies on copper. If you see a red precipitate, then you know that you have a reducing sugar in your sample. Now, very importantly, although this doesn't show up frequently on the MCAT, but just so you know, sometimes ketoses, which contain ketones, will act as reducing sugars. Now, this doesn't really make sense at first glance because we know that ketones cannot be further oxidized, so they're not going to act as reducing agents. Well, what's really happening is these ketones can be converted, can undergo tautomerization, in which case you'll have aldose or aldehyde intermediates, and those aldehyde intermediates, those aldoses, can undergo the oxidation process and act as reducing sugars. Okay, now let's talk about the esterification reaction. Very straightforward. So when you have a carboxylic acid and an alcohol in organic chemistry, they form an ester. So you can see it right here. So you have 3 carbons over here along with that oxygen and this oxygen as well. And then you're just sort of combining the 2 carbons of the alcohol with that, and that is your ester. It's very straightforward. Always make sure you count your carbons so that you're not missing anything. So this is essentially what happens with, what can happen with sugars because they contain alcohols. If you react them with a carboxylic acid, they'll undergo a Fischer esterification reaction. Now, this is kind of a special type of esterification where you have phosphate esterification. So as you'll see in our video on glycolysis, the cell will attempt to trap glucose inside of the cell in order to carry out glycolysis, and the way it does that is it uses ATP using an enzyme called hexokinase to phosphorylate glucose. And when you phosphorylate glucose, because of that negative charge on the phosphate group, it can no longer cross the plasma membrane, and therefore it's trapped inside of the cell. All this to say that this is another kind of esterification reaction that you can have with sugars. Okay, let's do some knowledge checks. I know that was a lot, but let's push through these knowledge checks over here. Which of the following best explains why beta-D glucose predominates over alpha-D glucose in aqueous solution? So you can pause, but I'm going to jump straight to the answer right here. So the answer is beta-D glucose contains fewer axial substituents. So recall the hydroxyl group was in the equatorial position in beta-D glucose, and therefore it was more stable. Okay, next we have a keto sugar gives a positive Benedict's test despite lacking an aldehyde group. Which of the following best explains this observation? So I'm going to jump to the answer, but feel free to pause the video. So the answer is ketosis can undergo tautomerization to form aldose intermediates. So recall that normally only aldehydes or aldehyde-containing sugars should undergo a positive Benedict's test, but because ketones can sometimes tautomerize into aldoses, so you'll have a positive Benedict's test. Finally, our last question here, 2 monosaccharides differ in configuration at carbon 3, but are identical otherwise. Which of the following best describes their relationship? So they have multiple chiral centers, they only differ at 1, so that's going to be a diasterer. All right, so that concludes our video for today. If you've found this video helpful and like the way I teach, I'd recommend watching the rest of the playlist. I spent a lot of time making sure that the videos build on one another, so it makes sense to watch them in order. Also, don't gatekeep these videos. If you have a friend who's currently studying for the MCAT, be sure to share the playlist with them. It really helps the channel grow, and I would really appreciate it. Also, did you know that every single video has customized Anki cards associated with it that cover the concepts that we discuss in a video? So if you want a way to be able to actively review the material and retain it for the long run, I've got you covered. If you're interested in accessing the Anki cards or even the slide decks within the videos, then check out our Patreon or reach out to me directly for alternative ways to access the Anki cards and the slide decks. Finally, these videos, at least as of now, are sponsored by you. So if you're interested in supporting me to be able to continue to make these high quality MCAT playlists, you can support me either via Patreon or by buying me a coffee. Links in the description down below, as well as the links for everything else, including the link to our growing Pre-Med Companion Discord community. Thanks for watching, and I'll see you in the next video.